we're going to talk a little bit about electroporation. It's a common non-viral method for NK cell engineering. And so, Bob, while I still have you, how does Celularity view electroporation as a method for genetic modification of placental-derived cell therapies?
I think we have a pretty pedestrian view of this, right? We see electroporation as a way to get the CRISPR-Cas9 and gRNA edits into the cells, right? And we're using electroporators to deliver siRNAs into cells, and we've been fortunate to look at the MaxCyte and the Lonza systems in our facility. And we think it's a really important tool for genetically modifying our placental-derived cells. And in our particular application that we focused on, we were looking at ways to avoid host rejection and improve persistence of our cells in vivo. So in that setting, in that application, electroporation was a very easy to understand, effective way to get the editing tool into the cell. And that's really the most simplistic way that at least I look at it.
Okay. Next question for you, Bob. Pretty high level, but still pretty important. What are the advantages and disadvantages you see in electroporation compared to other methods for your specific cell types?
I think electroporation has got a higher gene editing efficiency than that of the viral vector method. Since gene editing efficiency by viral vectors depends on the transduction efficiency, which is often really variable from experiment to experiment, from run to run, electroporation can be more consistent and can lend itself to a industrialized approach to making these products. Look, at the end of the day, it's about making these products at scale to a high quality, so that they meet high release specifications. And since NK cells seem susceptible to electroporation, and it's relatively transient in nature for overexpression of genes and could have certain disadvantages, we're still open to evaluating the tool, the electroporation tool. That being said, it's all about having a gene editing efficiency that is better than what we can get with just traditional viral vectors.
Sure. Okay. Paul, same level of questions along electroporation from Beam's perspective. So first up, we know that LNPs can enhance the efficiency of gene delivery in many ways. How does Beam Therapeutics view electroporation as a delivery method compared to other approaches like lipid nanoparticles or viral vectors for your base editing therapies?
Yeah, it's a great question, and I think Robert hit on a number of themes that I would agree with. In general, electroporation is a very universal approach, and you can get very high efficiency of delivery of nucleic acids into the cell through electroporating the cell membranes. That is very effective in an ex vivo setting. I think that has led to, and is a workhorse for us in a lot of our ex vivo programs, getting very high editing rates, which is along the lines of what Robert was saying, very consistent results with us as well. I do think, though, there is, depending on the cell type, a cost to electroporation, and we can see sometimes some cell types do not always have great viability after the electroporation process, and we have an impact on yield. And so in autologous programs, that can tend to be a big deal, where you're collecting cells from the patient and giving back to the patient. You really care about making sure you give as many cells back as possible. And electroporation can, in some cases, have an impact on that yield. And that's where it has brought us to LNPs in some of our ex vivo programs, because we can improve the yield and still get high editing efficiencies. But it does require, I think, a lot more optimization of the LNP formulation to achieve that. And that's really the trade-off, is can you get LNP formulations in the ex vivo setting that give you the same high editing efficiencies that you get with electroporation, and then also improve on the yield and viability issues that we sometimes see with electroporation for some cell types. And that's kind of the trade-offs and the type of work we do, like in pre-DC nomination stage, to decide on what's the right approach for a given product.
You know,
Gotcha.
I think Paul really hit the nail right on the head there. And I didn't want to come out and say it, but I agree completely. If the method you have to use in order to get the editing tool into the cell does some degree of damage to the cell and reduce its long-term viability, that has an economic consequence in producing the product, right? That's one of the reasons why LNPs are so exciting in so many ways, because if you can get your edit without damaging, and by the way, arguably, and Paul pointed this out, if it's an autologous model, right, where the number of cells that you have to work with is a rate limiter, you want to do as little damage as possible. You want to have the highest post-manufacturing viability that you can have. With placental cells, because of scalability, we don't think about that as much, but nonetheless, it's critically important. And so, from my point of view, and Paul put this perfectly, if you can have an array of tools to use, you're going to pick the tool that at the end of the day gets you the editing objective you want with the highest viability and the highest ultimate yield. That's what ultimately is where the rubber hits the road.
Sure, that makes sense. And perhaps this is a good time to address one of the questions that I noticed just came in from the audience, because I do think that this will piggyback on what you both were just saying. Does electroporation reduce viability of T cells? That's open to either one of you, whoever would like to respond.
So our T cell program, I can take it, and Robert, jump in if you would like. Our T cell programs, we use electroporation, and our programs are all allo approaches, so we're not quite as limited with the number of cells, and we have, there's always some impact of electroporation, but on viability. But if you optimize your pulse sequence, and you can do a fair amount of work to really balance the trade-offs between impacts on viability and high editing efficiencies, we've found great success that electroporation works quite well with T cells and giving us both high yields and high editing efficiencies.
And to add to that, the other thing, look, the field is evolving rapidly, and other factors will impact the sensitivity of a cell to electroporation, right? And I'm a big believer that the age of the donor cell, especially in the autologous setting, has an impact ultimately on how susceptible they are to long-term negative effects of electroporation, right? And we're still, the jury's not out on that, but when using cells derived from newborns, we think the membrane stability ultimately means that you'll have less long-term negative consequences of electroporation. That's my belief. That's my suspicion. And as we kind of learn that, it's going to help us select which platform to use for which particular approach. In other words, if you need to get a lot of edit in, and you need to punch a lot of holes to get those edits in, and that consequence is that you have a lower yield of ultimate product, that's going to impact your selection of the cell platform to use.